Okay. So it is an honor for me to introduce Professor Marlene Behrmann, who is a Thomas Stockham Baker University Professor of Cognitive Neuroscience in the Department of Psychology at Carnegie Mellon University in Pittsburgh. Marlene did her BA in Speech and Hearing Therapy, then an MA in Speech Pathology, and then a PhD in Psychology at the University of Toronto under the supervision of Professor Morris Moscovich. She held multiple academic positions and taught at multiple universities in the US and outside the US. She received dozens of awards honoring her scientific achievements, her mentoring commitment, her commitment to promoting science, promoting equality in science, and her dedication to voluntarily supporting scientific activities. And I'll mention only a few, as the Vision Sciences Society's David Teller Award, Exceptional Scientific Achievements, commitment to equity, and a strong history of mentoring, and the Faculty Member Award for Neuroscience of the F1000. She's a member of the National Academy of Sciences and a member of the American Academy of Arts and Sciences, just to name a few. She holds editorial positions in multiple journals, a member of many cognitive sciences organization committees, including funding bodies like the NAS, NIHS, NSF, and SFN. She published hundreds of papers, many of them in top leading journals as Nature, Neuron, PNAS, Trends in journals, and others, and also dozens of book chapters. Her research involves vision from a very broad perspective, including high-level vision as face, object, word, and scene-related abilities, attention, action in the healthy brain, and also in different disorders as in autism, visual form agnosia, prosopagnosia, hemispatial neglect, and others. And she uses behavioral methods as well as neuroimaging techniques. Without further ado, and although there's much more to say, which I haven't covered, I'd like to welcome you, Marlene, to our seminar. And just to say that Marlene has said that it's okay to ask questions during the talk. So feel free. You can also write things in the chat and she'll address them later. So welcome.
Great, thank you. It's really great to be here. Wish I were there in person, but thank you for the opportunity to talk. I can't see you and I can't see the chat. So Sharon is going to be my eyes for that part of it. I'd like to talk to you today about hemispheric organization, particularly in the visual domain. So let me start off by saying that to the naked eye, anatomically, the two cerebral hemispheres look remarkably alike. There are small differences, like in the patalia, in other words, the extension here in the right frontal lobe and here in the left occipital lobe. And this gives the brain a kind of a torsion that is known as the Yakovlevian torque. But you really have to look carefully in order to be able to see this. In contrast with the absence of overt structural changes, the popular press has made a good story about differences between the right and the left hemisphere. Here is one depiction, here is another depiction. This is kind of a hippie depiction with a little bit of wording. In all of these images, they portray the important property, and that is that there is binary segregation between the hemispheres. And I like this particular depiction because here you can see the bland, uninteresting left hemisphere. Everybody's in their gray cubicles, and the right hemisphere seems to be having all the fun. Some people are trying to cross over actually, but are not that good at it. And the question is, if we want to understand the nature of segregation in the two hemispheres, in the visual system in particular, where shall we look? So obviously we're not going to look in early visual cortex because it's essentially homologous. And I'm not even going to spend any more time on early visual cortex. We may well look at depictions like this, which show, for example, greater right hemisphere engagement in face processing and left hemisphere engagement in orthographic or word recognition. But in fact, we don't even have to rely on these popular press depictions because, of course, we can put people in the bore of a magnet and we can show them all manner of images, and we can examine the category selectivity here on the inferior ventral cortex. This is from one of our studies. There are thousands of these studies at this point in time. And mostly what I want to do is focus on two regions that have typically been thought to be quite distinct. And that is the visual word form area with preferential activation to word forms in the left hemisphere, and the fusiform face area shown here in red with selectivity for faces. There's actually good reasons theoretically for there to be the segregation of representations for these two stimulus classes. Firstly, the image properties of these images are so disparate, it makes sense that there may be two independent systems. It's also the case from an evolutionary point of view that whereas face recognition has been around for a long time, we've only been able to read, in other words, the general population has only been able to read for about 150 years. Prior to that, it was mostly priests and monks. And lastly, there are acquisition differences, so that whereas face recognition is acquired incidentally over development, reading is only acquired in preschool or in elementary school with hundreds of hours of practice. So I'm first going to show you evidence that supports this claim of segregation. I'm then going to challenge that by showing you data that really argue against such a binary segregation view. And I'm going to talk about the mechanism that gives rise to this not so segregated profile. And particularly, I'm going to argue that faces and words, which are thought to be unilaterally represented, share computational principles and are actually dynamically interdependent. Last, I'll talk about how plastic or immutable this hemispheric organization is. So let's start with the evidence for segregation. As I'm sure you all know, a lesion to the right hemisphere in the location of ventral occipitotemporal cortex, even just on the right-hand side, is enough to give rise to an impairment in face recognition. And pretty much in the homologous region in the other hemisphere, a lesion to the left hemisphere can result in pure alexia, which is impaired word recognition in premorbidly literate adults. And these neuropsychological data have been used to support this claim of independence. There are also data from evoked response potential. Here's from some early work, and I showed it to you because of Shlomo Benton, showing activation over the right posterior region for faces and over the left hemisphere for words at the N170 time. And many functional imaging studies, here from some early studies, the seminal one by Canwish et al., showing activation in the right fusiform area for faces and in words in the left hemisphere. So in a nutshell, across multiple domains, we see evidence of segregation. But if one looks a little bit more closely at some of the data, the evidence against segregation is actually quite overwhelming. So let me show you some of that. If you look at many of these studies where they look not just in a single hemisphere, you can see bilateral activation for faces. Here are a bunch, here is a schematic depiction, and here are a bunch of word studies, including some recent ones showing bilateral activation for words. In fact, in the very study where both words and faces are shown to the same individuals, and I'd like to share this particular study with you because it was done when I was on sabbatical at the Weizmann Institute with Rafi Malach, and it was just an incredibly awesome experience. So in this study, words and faces were shown to the same individuals. This is the inflated brain, and we're looking at the inferior surface of occipital ventral cortex. And on either side, you see the flattened brain. And what I'd like to point out to you is that in both hemispheres, there is both orange activation for faces and purple activation for letter strings. I would, however, like to point out that in the left hemisphere, there is more purple than there is orange, so greater activation for words than faces. and in the right hemisphere, greater activation for faces than words. I'd also like to point out to you that if you take the peak of the activation site for words and faces in the two hemispheres, in Taylor coordinates, these are roughly equivalent, suggesting considerable homology between the two hemispheres and suggesting that both hemispheres are activated by both stimulus types, but in a graded or weighted asymmetric fashion. Some recent EEG data that we collected, again showing words and faces to the same individuals, also show bilateral activation for words and for faces, roughly at the N170 time. Here are two different time points. Let me show you some other ways in which there seems to be equivalence between face and word recognition. There is a very slow maturation trajectory of activation for face recognition. This is work done with Susie Scherf, but Kalinikral Spector, Golije Golorai, very similar data, and many others do as well, showing the FFA both on the inferior and also looking at face activation on the lateral surface. We see a similar profile in adolescents, but children age 5 to 8, who are many of them at school already, are not showing on the group map face selective activation, so slow profile. We also see a slow maturation profile of the visual word form area. This is work by Guinevere Eden, where in adults you see, again, note bilateral activation, but in children age 6.4 to 11.6 years, which is really quite old, we're only beginning to see sort of the glimmers of the left hemisphere VWFA. Another way in which faces and words are really similar is that they can both tune this occipitoventral regions by experience. So this is the study from Isabel Gauthier and colleagues, training people to see greebles. And as you see here, over the course of training in the dark color here, you see an increased activation. This is the sum of the T values for greebles, and the open bar here is faces. I'll also point out to you, because I think this is so interesting, is that across the sessions of greeble training, there is a reduction in face selectivity, and a good example of competition and something I'll return to later. We see the same experience-dependent tuning in the visual word form area. This is work done by Chris Baker, where in English readers, you can see really good tuning for English words and letter strings. And in Hebrew readers, you see tuning for Hebrew orthography and also some for English. Brains aren't born into a particular language. And so clearly this is a result of exposure and experience. So although there is evidence for segregation, there is also considerable evidence for homology, as I've just pointed out to you. So there are these many common characteristics for face and word activation and its developmental and experience-dependent unfolding. And so I'd like to now discuss the mechanism by which this mature adult profile emerges. And in particular, I'm going to argue that faces and words, the mechanisms are interdependent and share computational principles. So we've already seen that faces and words are really very similar to each other and rather different between the two groups. One of the important claims, and this is particularly played out by Ifat Levi, Rafi Malach, and people who've done that kind of work, has been that each of these classes require fine-grained visual discrimination, given that the exemplars in each of these categories are so very similar to each other. And in fact, in that very same study that I had showed you previously with bilateral graded activation, here is the map just from a single individual in whom we also mapped out retinotopy. And I'd like to point out that in both hemispheres, the region of activation, here for letter strings and here for faces, is sitting not in retinotopic cortex, but just in the place that would be the anterior extrapolation of the fovea, were such a thing possible. So if the fovea indeed were extrapolated anteriorly, this is where these regions become activated. because fine-grained visual acuity is really necessary to be able to discriminate between these homogeneous exemplars. We also mapped out eccentricity, and as you can see here in green, these are eccentric areas, blue are the more foveal areas. These outlines in purple and in orange correspond to the group map of face and letter string activation. And again, you can see that in both hemispheres, these occupy central vision. Okay, so I'm going to put forward a hypothesis. I'm going to argue that to start off with, there's competition for neural positioning and representation for faces and words, given that they both need extra striate but fine-grained visual acuity. Over the course of development, I'm going to argue that initially there is no lateralization in the hemispheres, and both hemispheres can recognize faces. And in fact, we know some of this because of early work by Scania Deshona and her colleagues showed that children with a lesion to either hemisphere early on have difficulty in face recognition. The next step in this mechanism is that as children begin to learn to read, in other words, begin to optimize the left hemisphere for literacy, which entails the coupling of the visual areas together with language areas to keep connection lengths short, and this is the standard profile in modal right-hander. So by virtue of competition, do face representations be shunted out to some extent, not binarily, but to the right hemisphere. And so it's through this kind of jostling between face and word representations that we get this graded, weighted asymmetry. Okay, so I've already told you that there is this competition for the extrapolation of foveal regions. I've also suggested that by virtue of this representational cooperation and competition between homogeneous exemplars of these two groups, there is competition. And I first need to go ahead and show you that when we look at, say, face activation in the left hemisphere and word activation in the right hemisphere, in this graded view that I've been arguing, that those regions actually play a functional role in recognition. So the question is whether or not in the other hemisphere, say in the left hemisphere for faces, there is category selectivity that is functional. And so to evaluate that, I'm going to show you data from 4 pure lexic individuals with circumscribed left hemisphere lesions, 3 prosopagnosics with right hemisphere lesions. And I'm going to make the following prediction. Relative to controls, the patients who are alexic with a left hemisphere lesion should be super impaired at words, it's a big circle, and a little bit impaired at faces, and the converse pattern should be true in prosopagnosia. I just want to tell you that these individuals have no level visual deficits. We ran multiple experiments. I'm just going to show you 2 examples. The first one is to examine reading in both of these populations. The experiment is simple. A word appears centrally. They simply have to read the word aloud, and we measure accuracy and reaction time. Relative to matched controls, you can see the alexic individuals, this is the percentage of errors, make very many errors. They actually make more errors as the words get longer, which is a standard profile. But I also want to point out to you, relative to the controls, the prosopagnosic individuals are also impaired. And so we already have one side of this equation as predicted. Let's look at these very same individuals and look at their face recognition ability. Here's one experiment. 2 faces appear on a screen. They can be completely different, face A with face B, or they could be 2 of the very same faces and participants are making the same different judgment. We had 2 other conditions when faces were different. And the one is that we morphed face A with face B. So 66% face B, 33% face A. Let's call this the intermediate condition. And the difficult condition is where face A is paired with another face that's 66% face A and 33% face B. So the idea here is to try and draw up a parametric function of face discrimination ability in these two neuropsychological populations. Okay, so here are the controls. Notice that the difficult condition is so difficult, even the controls are performing pretty poorly. So this may not be the most sensitive condition, but relative to controls in the easy and medium conditions, of course, the prosopagnosic individuals perform poorly. And interestingly, the alexic individuals also perform poorly, actually almost as poorly as the prosopagnosics, except perhaps in the easy condition. So the data from this suggests that the bilateral activation that we see in imaging plays a functional role. This is not just the epiphenomenal cascade of information across the corpus callosum. Okay, so having shown you that both hemispheres are involved in both face and word recognition, I want to just spend a minute and show you some recent data that we've collected, which is essentially a redo of the Hassan Malach study that I showed you earlier on. But specifically, we want to examine the competition and the interdependence between face and word recognition in the two hemispheres. This is a group map. Words are in blue, faces are in red. It's basically what we've seen previously. What I did want to show you is that there is a correlation between face activation and word representation, so the extent of the activation in the two hemispheres. Kind of as I had suggested, there is this jostling for neural position with the consequence that the better you are at word recognition in the one hemisphere, the better you will be at face recognition activation in the other hemisphere. Okay. But there's a little bit of tension in what I've shown you so far, because I have told you that there is bilateral activation. But if that were true, and not only is there bilateral activation, but both hemispheres play an active role in perception. And if that were so, why would you ever get a neuropsychological patient with prosopagnosia? Why doesn't the other hemisphere simply just step in and take over the function? So we examined the individual profiles in this group of participants. We've scanned 28 so far. We're scanning up to 52. Here are two instances where you can see the bilateral profile quite nicely. But I'd like to show you two individuals who show very strong unilateral profile. This person pretty much has got face recognition just in the right hemisphere and word recognition in the left hemisphere, and this other person does as well. And my hypothesis is that these people in the bottom row, if they have damage later on, are the future pure lexics and prosopagnosics. And in fact, these disorders are very rare in the population. And so my guess is that these are kind of in the tails of the otherwise normal distribution of bilateral representation. Okay, so where would this individual variability come from? Like, what determines who has a bilateral or a unilateral profile? In addition to the functional imaging, we've also got very detailed white matter imaging. And the hypothesis here is that one possibility has to do with the nature of the white matter connectivity between hemispheres. So the claim is that the smaller the volume of the splenium, which connects visual cortex, the more lateralized these activations will be. So if you've only got a tiny region that connects the two hemispheres, it's better to have word activation in one side and face representations on the other side, because the hemispheres don't talk very much. And the contrast here is the genu of the corpus callosum. And the greater the splenial volume, the more bihemispheric the representation will be. I don't know whether this is true or not. We only have partial data. This is the hypothesis. It's also possible that the results come not from between hemisphere, but from the within hemisphere connectivity. And we have very detailed data on the inferior longitudinal fasciculus, as well as the arcuate fasciculus and other relevant fasciculi. So either the between hemisphere thinning gives rise to lateralization, or very strong within hemisphere connectivity can give rise to lateralization. So stay posted, and hopefully post-pandemic, I'll be able to tell you the result. Okay. Let me turn now to this question about where this topography comes from. In addition to these individual differences, how does it emerge over the course of development? So I've already shown you an instance of a hypothesis which argues that there is this competition for representation that only starts up once individuals start learning to read. And by virtue of the left hemisphere optimization for word recognition, we see consequent lateralization of faces to the right. And this is contingent on the nature of the between and within hemisphere connectivity. I'm going to show you some evidence, because essentially the story that I've told you is a developmental story over the course of acquisition of word recognition. So here are behavioral data from adults, adolescents, and children. The experiment is simple. There's a fixation point, a target, in this case, face comes up for 750 milliseconds, long enough for the younger children to be able to encode it with precision. Brief fixation, and then with equal probability, a probe face appears in the left or the right visual field, and participants are making a same different judgment, and the keys are randomized. There are both blocks of faces and blocks of words. And so the design has two fields, two classes by age. This is the accuracy profile on the y-axis for adults. And as you can see here, and is the standard pattern, there is greater accuracy in the left visual field, i.e. the right hemisphere, over the right visual field, the left hemisphere, for faces, and the converse for words, i.e. better right visual field discrimination for words than the left hemisphere. So this is the standard complementarity of the two hemispheres. I do want to point out to you that in, let's call it the non-preferred hemisphere, so the right visual field for faces and the left visual field for words, accuracy is about 80%, consistent with this idea that there is functional engagement of the hemispheres. The young adolescents show roughly the same profile for words, greater left hemisphere accuracy for words than right hemisphere, but no lateralization for faces. And if we look at the youngest children, whose accuracy overall is much lower, we again see no lateralization for faces, but the glimmerings of lateralization, in fact, it's significant for words. And so we see this early emergence of lateralization for words. And the claim is that it's this lateralization that drives the emergence of lateralization for faces. So we looked at that specifically by correlating the degree of lateralization, so the accuracy for faces, along with the, on the x-axis, performance of these individuals on a standardized reading test that has got various measures. I'll also note that we partially correlated out IQ, other perceptual abilities, etc. And even when that was all done, we still see a positive correlation, so that the better the reader you are, the greater your degree of lateralization for faces. This is true in the young adolescents and the children. It's not absolutely contingent on age, it's contingent on how good a reader you are. So here we've got some kids who are pretty good readers, well within the young adolescent group, who are also showing the same face lateralization. We collected ERP data in a new group of adults and just the children. It's exactly the same experimental paradigm. We are only looking at the amplitude in response to the presentation of the target. So just the central face or the central word, unconfounded by the motor choice responses that they make. So here in the adults, I just want to tell you amplitude, this is an N170, it's a negative deflection, but to make it consistent with the figures I've just shown you for the behavioral data, I've plotted negative upwards just for convenience. So here we see greater N170 amplitude over the right hemisphere than left hemisphere for faces, and over the left hemisphere than right hemisphere for words, really consistent with what we've already seen. And in the young children, these are about I think the mean is about 7, we see, again, significant lateralization for words, and only the emerging trend, it's not significant, of face lateralization. So the claim here is that faces and words are interdependent. The better you are at word reading, the more lateralized you will be at face recognition, taking into account the hypothesis that the connectivity of the white matter plays a role as well, an as yet to be determined role. Shall I stop and let people ask some questions?
As you please, sure.
If anybody wants.
Yes, I have a question, Marlene.
Rafi, yeah.
Very, very interesting. I wonder, you have a very straightforward prediction, and that is that illiterate individuals should have equal representation, and more when they learn to read, you should start seeing a shift.
Great.
I wonder whether you have any.
Yes, I can answer that very well. The first thing I can tell you is that this was a study that I had proposed to NIH, I don't know, 10 years ago. Because in Pittsburgh, the State Department has resettled refugees from the Sudan and Ethiopia. These are mostly mothers and children. The fathers have all been killed in these just horribly devastating wars. And in a partnership with the Literacy Council in Pittsburgh, with interpreters and everything, we proposed the study to NIH. NIH came back and told us that we could not do the study because we had proposed to pay these participants just like we pay everybody else. And that was unduly coercive for this impoverished population. And B, we couldn't guarantee that these people could give informed consent because they may not have a full appreciation of the scientific issue. So we argued, we went to the ombudsman, we failed. But Stan Dehane, and actually more recently, Carreras and colleagues in Portugal, who I think ran the study in Mexico. So 2 groups have run this exact same study. I refer you to the Dehane study, which was the first published one, which does exactly as you ask, Rafi, and that is it shows that in illiterate adults, you get bilateral face activation. And as individuals become increasingly literate, even in adulthood, you begin to see activation in the visual word form area and a reduction of face recognition in the left hemisphere. They didn't show what happens in the right hemisphere as literacy is acquired, unfortunately, because of course the claim is that you would see an increase in face activation in the right hemisphere. We just saw a reduction in the left hemisphere. But it's a completely great question, and I think the studies have been extremely well done.
Can I just ask?
Go ahead.
In the experiment that you've just described, if you had a level of difficulty that was different between the children and the adults, which you've got, and you've got that in your results, and you didn't match them for difficulty across the words and the faces, you could get that differential response, couldn't you?
So if I'm correct, this is a case on this graph where children and at least the young adolescents, and maybe the adults as well, perhaps, are matched in terms of their reading profile.
Yes, so, saying you have to match across the face differences and the word differences to make sure it's the same level of difficulty. Otherwise, it could just be that faces are more difficult to recognize and discriminate than the words.
So you're absolutely correct. We have that analysis. I don't have it here, which is why I showed you this one, which was my best effort at going in the direction that you were hoping for. So what we actually have done in a recent study is that we have used words for the young children that are titrated to their age level for reading. So if you're 6 years old, you read 6-year-old words, and if you're adults, you read adult words. And face representation, well, that's actually difficult to do matching. Our best effort has been to make faces more different for the younger children and less different for the adults, to equate. It's essentially, you need to equate the playing field in order to see the nature of the cortical activation. And our more recent data, which does that, nevertheless shows that the better you are at word reading within an individual. So it's equated, discrimination is equated at the sample level, but there are still individual differences.
Individual differences, yes.
Yeah, and that individual difference still plays out in roughly the way that I've been describing here.
Yes, thank you.
Yeah, thanks, good question.
Marlene, could I ask a question, please?
Hi, Avital.
Hi. So I'm intrigued by the data you showed that people with pyralexia or prosopagnosia show an equal, well, a similar deficit in both recognizing faces and words.
Yes,
so I was wondering,
not equal in severity.
Yeah, but similar. So I was wondering, is this really the result of the actual injury? Or could it be that because these two processes have some shared they share some mechanisms, some brain resources as we grow older, could it be that plasticity in the intact hemisphere is now in some way the other component, be it face recognition or word recognition, is now exposed, and these resources are now shared between these two behavioral needs?
It's an interesting question. Where I get stuck with that, or sort of come up against a wall, is understanding what these brain resources are, and these elusive brain resources that you know, and it's an argument that is made very often. It's sort of ubiquitous everywhere you look. It's either attention or it's resources of some sort. So for me, in the absence of a clearer understanding of what that is, I'm reluctant to endorse that.
So actually, my question is more specific. So given a brain area that once was in charge of both facial recognition, so a territory that was shared when we were children, perhaps, and now we lose the other homologous area.
Yeah.
Could it be that by virtue of plasticity, now the intact face recognition area would process words as well?
Yeah.
And now for this reason, we would see a deficit in behavior? So
yes, now I understand better what you're asking. Okay. I don't know. I'm going to show you some data now about the plasticity of the system that may go some way to addressing your point, but we can return to it at the end.
Sure. Thanks.
Okay,
can I ask a question?
Yeah, go ahead.
So would you say that individuals that are more lateralized should be better at reading and face recognition, or not necessarily?
I don't necessarily think so. I think that systems can be optimized. So there may be a sufficiency argument here. So as long as, for example, the left hemisphere has good coupling between, say, these posterior extrastriate regions and language regions to some extent, so sufficient coupling, that may be enough. It may not be that you need to be extremely lateralized for this to happen. You can be bilateral, but as long as your left hemisphere is well coupled, well connected, that may be enough. It's a good question. I don't know the answer to that.
Thank you.
And I'm going to write it down and look at it more.
Is there time for one more question, or shall we go?
Yeah, go ahead.
Sorry, Marlene, really interesting data and hypotheses. I'm just curious, how broad do you think this mechanism of competitive representation could be in the brain, in the sense that would it expand only to areas that are for newly learned skills, either throughout the lifetime or in evolutionary scales, or do you think that that's something that happens developmentally for other things like others across senses or across computations more generally?
I'm going to show you a slide towards the very end in which I'm going to reference other people's work, which argues for a very similar kind of competitive mechanism. So I'm going to go out on a limb here and say that I think this is a ubiquitous, maybe even universal computation. That's how brains become sculpted by virtue of collaboration and competition of neural representation. And this could apply across modalities, across information domains. And I think, may apply in some of your interesting work as well. Great. All right, let me go on and show you the last little bit, which has to do with plasticity or immutability. So I'm going to show you data now from children who have had one hemisphere removed in the extreme. So these are cases of hemispherectomy, or as is often called in these cases, hemispherotomy, where a little bit of frontal cortex and a little bit of occipital cortex is preserved so that the hemisphere doesn't kind of move over entirely in the skull. So these are sort of anchor points, but essentially the right hemisphere has been removed. And what's interesting about these cases is that notwithstanding this dramatic resection of brain tissue, their behavior is reasonably well preserved. But a single case like this in adulthood, and this is one of the prosopagnosia individuals I showed you earlier, a circumscribed, pretty focal lesion like this in the right hemisphere in adulthood renders you prosopagnosic. And there are not many reports of recovery from prosopagnosia in adulthood. So children present to us a unique opportunity to study plasticity. We've scanned 23 such individuals. On this side is a subset who have resections that encompass the ventral occipitotemporal cortex. We also have behavioral data, which I will show you. And we have controls, both who've got resections outside of ventral occipitotemporal cortex and matched normal controls. Just to show you, we also have ECOG data from these individuals, but just in the last 2 weeks have collected stereotaxic EEG recordings with deep intracranial electrodes. And so I'm excited to look at those data. Temporal resections are most common, or temporal lobe epilepsy is the most common type of epilepsy. And so for somebody like me, even though this is somewhat macabre, this is a good thing because we have lots of cases. So this is an individual who then went on to have the resection, and here is the resection. We've scanned these individuals cross-sectionally and longitudinally. We never see any resolution in the hemianopia. There are occasional reports in the literature about resolution of hemianopia, so that a single hemisphere represents both the ipsi and the contralateral field. As far as I can tell, the damage in those cases is prenatal, or these kids are born with something like achiasmia. So we don't see a resolution. So this individual has got a blind field, blind left field, 3 years apart, as shown here. We always do Goldman perimetry in these cases. Just to show you a little bit about the performance, we do face recognition. This is a CFMT for children. We do an object matching task. They could be the same, or they could differ at differing levels of similarity. So it's sort of perceptual challenge. We also check intermediate level vision. So this is the contour integration task. An image of Gabor's is shown like this. They don't see this, but the Gabor the circle, well, the ellipse of the Gabor could be tilted a little bit leftward or a little bit rightward. And the kids are saying if it's a little bit more left or a little bit more right. We also manipulate the alignment of these think of them as sort of beads in a necklace. And obviously these red dots are not present. And we can, and we obtain thresholds. And the lower the threshold, the better they are at doing contour integration. We also run the sort of well-established glass pattern by Terry Lewis, Daphne Maurer, where the participant has to say in this interval, which has more swirl. One could have 100% signal, this has got 50% signal, one comes before the other in sort of standard thresholding, and again, lower is better. With two exceptions, all of the individuals we've tested so far perform within normal limits, both in the pattern recognition and in global form perception. And the two exceptions are an individual like this, who is born with polymicrogyria. I hope you can see how many gyri there are. That this individual has epilepsy and goes on to have surgery does not remedy the widespread polymicrogyria. The other individual has widespread cortical dysplasia. Again, the brain is just very abnormal in its structure, and so surgery again may manage the seizures, but is not going to give rise to any cognitive improvement. Okay, our first hypothesis was that the reason that behavior may be so good might well have to do with these fasciculi, these white matter tracts that I already described previously within hemisphere inferior longitudinal fasciculus and fronto-occipital fasciculus. I'm not going to go into a lot of detail. Suffice it to say that we have a detailed structural imaging here, anterior section cases, posterior section cases, controls on these two different tracts, very carefully evaluated with a hypothesis being that there may be an increase in myelination or some other microstructural property in the resection individuals. Here are the data. The gray squares here is the region of the resection, and orange is contralesional, blue is ipsilesional. These are the controls, so as you can see, in fractional anisotropy, axial diffusivity, and radial diffusivity show no difference between the two hemispheres. And the patients actually show no difference between the two hemispheres as long as you are outside of the resected region. Here is another example. So it's not that there is either degeneration in the remaining tissue in the affected hemisphere. It's also not the case, as I will show you in a moment, that there is any change in any of these microstructural properties. So this is in the ipsilesional and contralesional hemisphere. These are the T-scores of the contrast taken from the controls, anterior and posterior. And at the bottom here are just 5 examples of patients. And you can see that with a few exceptions, the patients all fall within the normal distribution. And when they are outside, it's not a single patient is outside on all of these measures. And sometimes the outside, they're above the controls, and sometimes they are below the controls. Actually, my screen thing is in the way, so I can't even hear, but some below the controls. Okay, so I don't think that it's white matter that's doing the trick here. I also wanted to just share with you quickly that we've managed to conquer a number of methodological problems in dealing with these very complicated and abnormal brains. And this work has been done together with Daniel Glenn, who's a computer engineer at NIH. And if anybody is interested in how to align really odd hemispheres so that you can do things like segmentation of gray and white matter, et cetera, use fruit, HCP, whatever, free surfer. I'm happy to tell you how to do it. This is just to show you that this single individual's brain, once you put it through this, essentially what we do is we mirror the preserved hemisphere onto the missing hemisphere, and then free surfer and all these other automated programs work just fine. So if that's of interest, this is a side note. And now I just want to tell you, this is a quick side note to the side note, because we've just published a paper this week in Nature Communications in which with just 3 minutes of high resolution EEG data, we can localize the region of silence, i.e. the resected region. Here are 3 different individuals from this resection pool. This is the ground truth from the MRI scans, and we compare our algorithm called silence localization to most of the other sort of standard source localization, slightly adjusted so that we can look at the silence region. And we can match the ground truth much better than any of those. So if anybody is interested in this, I'm happy to tell you more about it as well. Okay, I want to, in the last few minutes, show you some group data on analyzing the functional properties of the remaining category selective regions in these resected individuals. And so I'll just remind you of this map. So we did a standard category localizer. Here we see the activations for faces minus houses and words minus faces. I'll just point out one to you. This is an individual with an anterior temporal lobe resection. This is faces are in blue, faces are in orange, houses in blue, looks pretty good. Words and faces looks pretty good. But let me quantify this for you. So there are roughly 9 regions of interest here. This is right and left, posterior to anterior, early visual cortex, LOC, the visual word form area, PPA, FFA, PPA. Okay, and that's all the regions over here. The analyses are always done by taking the T-scores of the controls. And I think there are 9 patients here, and each dot is a single patient. You can see that the patients almost without exception, maybe without, oh, here's one exception, where the VWFA of this individual is just out of the control. No, it's still in the control range. So they look absolutely normal. This is done on a permutation analysis by shuffling the labels, and you can see the groups are essentially within the normal distribution. We also looked at the correlation of the spatial position of these category selective regions, because of course it could be shifted to some completely different part of cortex. So we put a dot over each of the peak of the coordinate of the category selective regions, again for the 9 ROIs in those individuals in whom we can identify 9 ROIs. In this individual, we only get a subset of the ROIs. Oh, sorry. I seem to have not had, I don't have the actual slide here. It may be shuffled, but suffice it to say that if we look at a correlation of the 3D coordinates of the regions of interest in the patients where the regions exist compared to the controls, there is no difference in the spatial position. And the last thing we did is look at representational similarity using sort of the standard correlation matrix of sort of within and between. So this again is the RSA, the representational similarities T-score of the controls. Again, in all of these regions, the participants are falling within the normal range, except for these 2 individuals in the visual word form area. And it's because their visual word form area is actually in the right hemisphere. And I'll show you more about that in a minute. And also by permutation, the RSA looks similar. So what is being represented in the remaining tissue appears to be very similar in the patients and the controls. Okay, so as far as plasticity, we seem to see structure preserved outside the resection. The ROIs are preserved in the magnitude of the selectivity, in the spatial coordinates, and in the representations. I'd like to, in the last few minutes, zero in on 2 individual case studies that will demonstrate the computational principles that I've just described to you in terms of the interdependence of faces and words, but in a single hemisphere. Okay, so here is a single case. He's got a right hemisphere resection. You can see that he had a tumor here. This is the timeline of the longitudinal study. His resection was at age 6.9, and we scanned every 6 months after that. He was hemianopic for the duration. This is a schematic for you of the topological organization of the 2 hemispheres. So in this individual who doesn't have occipitotemporal right hemisphere, we're going to look at faces, which are in red here, and words, which are in brown, and their emergence over time. So to do that, we put a sphere over this ventral occipitotemporal region. The fusiform gyrus is here in pink, and occipitotemporal sulcus is in brownish. And we look at faces and words. This is across 4 of the scans from age 7, 10 to 10.10. Each individual dot here is a voxel in this sphere, as you can see, X, Y, and Z. The redder a voxel is, the stronger the face selectivity is, and the more blue it is, the higher the word selectivity. So notice that early on, there's no real strong commitment to faces or words. It's all kind of hovering at around the midpoint here. But over time, as this kid grows, you begin to see something of a separation, so that at age 10, there is a region that has more face selectivity and a region that has more word selectivity, basically abutting each other. So this is an instance of the jostling for competition over the course of development and eventual resolution of neighboring regions, probably not exclusive to one category or the other. Maybe what I should do is I should also plot both of these at the same time, like relative to a control condition, which I will do next time. And there is a significant change across all of these times. But maybe that's not so surprising, because we already know that you should see face and word selective activation in the remaining left hemisphere. What about an individual who has a left hemisphere resection? It's rather uncommon to see a visual word form area in the right hemisphere. You see it in some cases. It is bilateral, but it's not as strong and it's not as evident across the population. So I'm going to show you data from this individual, where we put the sphere here over the remaining right hemisphere region in exactly the same way. We've only got 2 post-surgical scans. Oh, and in most of these cases, we've also got pre-surgery as well as post-surgery. But I just wanted to show you these 2 post-surgical scans, where again, we see a kind of jostling for position between scan 1 and scan 2 significantly differently, with something of a greater commitment to face selectivity and to word selectivity in adjoining voxels in this 3D sphere. Okay, so let me sum up now by telling you that in contrast to the evidence for segregation, we see considerable evidence arguing against this neat segregation, this binary separation, and an account of interdependence, one of dynamic emergence with competition and collaboration for neural representations and possession, may well be the triggers that gives rise to the mature profile. And lastly, there seems to be considerable plasticity in this hemispheric organization, as we see in these cases. And actually, to return to the question that Rafi Malach asked earlier, we also saw that same kind of plasticity in the Dehaene paper, because he scanned adults who were illiterate. And you even saw the sort of plasticity and alteration in the functional activation profiles in adulthood as they became increasingly literate. So there may well be greater literacy, greater plasticity in childhood, but there still seems to be some in adulthood. And this is in response to Ella's question. This idea of there being this emergent dynamic hemispheric organization is also emerges in 2 other fields. This is in the anterior temporal lobe. And for those of you who are interested, I recommend that you see some of the work of Grace Rice. And there's a review and meta-analysis about how in ATL you see similar kinds of mechanisms at play that I've just described. And Otto Carneth, in talking about language versus spatial representations in the right and the left hemisphere, also articulates a similar claim, and that is, as language becomes increasingly sophisticated, spatial representations are shunted to the right hemisphere. So I'm going to end off here by saying that there isn't quite the binary segregation that the popular press, at least, would have us believe. And in fact, a better way of thinking about it is in terms of graded independence, where both hemispheres seem to be having quite a lot of fun, although with a slight weighting or asymmetry between the two. I'm going to end off here by recognizing the contribution of many graduate students at CMU over time, postdocs at CMU over time, a collaborator, David Plout, Jason Fu at NIH, and actually Daniel Glenn at NIH, and Christina Patterson, who is the head epileptologist at Children's Hospital in Pittsburgh, and with whom I have an amazing collaboration. I'm very grateful to her. So thank you, everyone. And I'm happy to take more questions if you wish.
Thank you very much, Marlene, for a wonderful and inspiring talk. And I'd like to ask everybody to unmute yourself and give Marlene a big thank you and applaud. And of course, everybody is welcome to ask questions. I have a few of my own, but I'll wait.
Okay. I'm not going to stop my slides in case I have to go back to.
Okay, sure.
To Sam.
If no one's jumping fast enough, I'll go ahead and ask. Beautiful data, really. I have to wonder about those individuals who had their hemispheres removed, that you've been tracking their development. Mm-hmm. Can you see, or do you see any differences in the connectivity of the areas that would become face or letter selective prior to the operation, or at least prior to the greater specialization of these regions?
I cannot answer definitively because we have not fully processed all the pre-surgical data. And again, we have both tractography data as well as functional data, structural and functional data. I will just caution that we may not be able to evaluate the question that you want, because even in these kids before they go to surgery, because the region is already dysfunctional and seizing, which is why they're going to have it resected, there may well be pre-surgical changes already. So we can't look at the change from a completely normal brain pre-surgically to an atypical brain afterwards. So I can't definitively answer. What I can tell you is that post-surgically, we aren't seeing massive changes. In fact, it was my hypothesis that we would see greater connectivity post-surgically, but neither at a macro nor microstructural level do we see anything like this.
Thank you.
Can I just ask, have you seen any children who've had their operation, their surgery at an very early age? We saw two, for example, that were very under 1 year, a long time ago. But we're looking at rather simple ideas, not looking at face perception.
Right. So we actually have, and I'm just going to stop the share for a moment. We have, so one sort of change in the zeitgeist these days is that these kids are coming to surgery earlier and earlier. It used to be the case that surgery was not done until they had tried 4 different pharmacological interventions and failed all of those. And then they insert stimulators, kind of like deep brain stimulators, that actually suppress the seizure activity. And they have to have failed those. And only then can they come to surgery. But that already takes years of where they could be developing normally while they go through these failed intervention attempts. And so given that the surgery seems to be so successful, surgery is taking place earlier and earlier. And of course, the earlier it takes place, probably the better the outcome. So we have probably 6 kids under 1 year of age. We have another bunch between 1 and 4 years of age. These kids are really hard to scan functionally. I had some experience in the past with scanning infants while they were asleep. And actually, Ilan Dinstein and I together with another group, had looked at kids who may go on to be autistic, but scanned them when they were infants asleep in the magnet. And we basically looked at functional connectivity of sort of superimposed regions, like we guessed where the language regions are, and we looked at the interhemispheric connectivity relative to kids who were not going on to be autistic. So these were like the baby siblings of kids who were autistic. So actually, we could potentially do something like that, is look at the brains of these very young kids while they're asleep. We've tried with 4-year-olds. One kid was trying to get out of the magnet during the scan, like busy taking head coil with him off the table. So that was a disaster. We haven't had a lot of success scanning very young kids. I think our youngest is age 5.
It wasn't meaning that you should scan them then. I was meaning because of the ability of very young children to make face discriminations.
Yeah.
Had a lot of experience of faces, but they haven't had necessarily a lot of experience of words or even picture books by the time they're that age. Yeah. So even if you scan them later, you find out about whether the early plasticity would give you a better outcome for faces than for words, or even when they were learning to read, it would lag behind. That's what I was thinking.
Yeah, it's a great question. It's a great hypothesis. I mean, in a related fashion, we know something about cataracts in young kids and about the impact of even, very early transient impact on the visual system. I'm actually working with somebody and we're looking at kids who are born without corneas and then who have corneal transplant and what happens under those kinds of circumstances, very early corneal transplants. So similar kinds of ideas about very early abnormalities and exposure to a particular type of stimulus and what those consequences are. That's a great question, thanks.
And I could also add that you should look at acallosal people for your callosal question, because, I mean, there again, they have had those abnormalities, usually from birth. And so they'd be another test case for when you're exposed to faces and when you're exposed to words.
Yeah, great. I've only ever tested and scanned a single callosal case. There are also individuals with callosal agenesis, not even a callosotomy.
Yes.
These days, a callosotomy is not performed very often because, of course, you would continue to seize in the one hemisphere. And so the cortical resection is kind of the surgery of preference at present. But yes, along the same lines, your question is really a good one. You're on?
Marlene, I'd like to ask one question that I have is with respect to the cytoarchitecture composition of these regions, because we know from a Kalanit study with the Ulick Center that there are differences in the psychoarchitecture of the, let's say, the fusiform subregions. I don't know if they have compared the right and the left, but that would be interesting to see if there's computational differences. And also I was wondering about the idea that whether you think there is something to the idea that the right hemisphere processes more things that are physically present in the world, whereas the left hemisphere is more, I know, because I'm thinking about 3D versus 2D. For example, I mean, when I compare text and faces to an object, and so, but it's not that, it's just an idea.
So, Sharon, to start off with, I think your reference of the Kalanit and the Ulish work is really a good one. And it may be the very reason that we see the emergence of face and word selectivity in the correct regions in these kids. And that is because of the cytoarchitectonic constraint. Like you can't go and develop face recognition in some other part of cortex because you don't actually have the cytoarchitectonic predisposition. So I think that's very relevant. As for, and I don't know whether they did the right and the left hemisphere. I usually always look at this, and my guess is because I haven't encoded it, they may not have, or it may not be different. But I'll go back and look at it. As for a 3D versus 2D, I think this is a great question. I mean, of course, we're scanning and testing 2D images, but we do know that real world instances of an object do in fact have differential processing to contrived 2D images. And I've done some work with Jackie Snow in individuals adults with visual agnosia, who are less agnosic for real world size compared to artificial, and also real world objects compared to their 2D counterparts. So this is a fundamental confound between words and faces. It would probably be really good to find another stimulus domain if we could. So I don't know, like if I think about the Greeble work, they were only 2D. They were only ever trained on 2D Greebles, but you saw the activation in the right hemisphere. So that may argue a little bit against this 2D, 3D split. But I have to remain uncommitted. I'm not committed one way or the other. And it's intriguing.
Yeah, just an idea. I'll think about it more. I'll come up with. Thank you.
Do you think that developmental dyslexia may reflect any initial differences in the symmetry or organization of the face areas?
Maybe I have the slides. Maybe I kept them in the back. Maybe I didn't. Okay, I actually, just to start off with everybody, this is, oh no, I didn't mean to do that. Sorry. I want you to, I actually do have the slide of Stan DeHane's work. Sorry, I'll come and answer your question in a minute. I really just want to show you. This is the DeHane study with the literate and illiterate individuals, and you can look it up. And what's super is that there's no visual word form activation in the left occipitotemporal cortex in the illiterates, and a lot of face activation. And as you become increasingly literate, so you see an increase in letter string activation in the left hemisphere and a decrease in activation to faces. Okay, so we ran the experiment that you're asking about in developmental dyslexic individuals, actually just the behavioral experiment, which is this one. And without going into all of the details, we looked at the prediction is that, let me make the prediction. If in developmental dyslexia, there is a problem in, let's say, phonological processing. So the language representations in the left hemisphere are not quite as they should be. There will be much less coupling between visual areas and language areas because they aren't, language area, phonology is not working so well in the left hemisphere. As a consequence of that, so reduced coupling in the left hemisphere, we should see reduced lateralization of faces because there's no competition to push faces out. The left hemisphere is not becoming optimized, and hence there is less competitive need for faces to be shunted out. I hope that that's clear. So that's absolutely true in developmental dyslexics. We see, we don't see as much face lateralization. These are adults with developmental dyslexia, by the way, compared to controls. We also, in the same experiment, looked at congenitally prosopagnosic individuals who are now adults, who are not that good at face recognition. But in that case, we fully expect the left hemisphere language regions to be intact. There's no reason why they shouldn't be. And so the coupling in the left hemisphere will be really strong. And so we should see lateralization for faces in the congenital prosopagnosics and lateralization for words. And we do, and others have as well. So I hope that answers the question.
It does in a way, but what I was wondering, I mean, you were suggesting the initial problem with dyslexia would be in phonological processing, and that's a very widely held view.
Yeah.
I wonder if differences in the organization of the face system might themselves be a precipitating factor.
Yeah, I can't answer that. We only ran this one study. Developmental dyslexia is a bit beyond my pay grade, I think they call it. It's entirely possible. As you well know, developmental dyslexia is a very complicated, intricate, and controversial domain. And there are probably many ways.
Thank
you. Maybe one more small question on my side, last one, I promise. You've shown a lot of very convincing evidence about the competition in terms of selectivity. Do you have any evidence about talking about the representational content of these regions and whether there are differences otherwise between right and left hemispheres for these domains?
Oh, I guess you can't see this because I'm not sharing that. I'll just try and share this. This is the representational similarity in the lobectomized, hemispherectomized individuals. But you may be asking about normal participants, is that so?
Yeah, and also for different aspects within the category, not across categories.
Yes, I can't answer that. As I mentioned, we've scanned 28 typically developing individuals already in this experiment. And where we have these individual differences as well. And we have some RSA data already analyzed, but not fully. And your question is a good one. And I will be able to answer both within and between category representational differences, hopefully soonish.
I'll look forward to that. Thank you.
Okay, so I'd like again to thank you, Marlene, for joining us. It was a pleasure having you. Exciting and amazing results. And I invite everybody again to give a big applaud. And thank you so much. Next week, I can't hear you.
I was going to say thanks for the opportunity. And it's totally awesome to see so many friends on the screen. It's like really lifted my heart for today. Thanks.
Good. And you did the same to us. So next week we'll be here at the same time again, Eastern time 8 a.m., Israel time 3 p.m., UK 1 p.m., and with Galit Yovel. She'll be talking about faces and I think artificial networks, but again, I'll post the precise title soon. So thanks again for joining everybody. Thank you,
Thanks, Marlene. It was great.
Thank you.
Thanks, Rafi.
Thank
Hi, Joe.
Marlene.
See you. Hi, Galia. Bye, everyone.
Bye.